Non-striated or smooth muscle cells are a major component of hollow organs such as the alimentary canal for motility, airways and vasculature for conduit, and urogenital tract for storage. Smooth muscles are generally divided into two major types: the multi-unit or the unitary, or the single-unit smooth muscle. In multi-unit smooth muscle, each muscle fiber operates independently of the others and is often innervated by a single nerve ending. Multi-unit smooth muscles are capable of finer control. Indeed, they are found in the iris, ciliary body of the eye, and the piloerector muscles of the skin. On the other hand, unitary smooth muscle means that a whole mass of hundreds to millions of muscle fibers contract together as a single unit. These fibers are usually aggregated into sheets or bundles, and their cell membranes are adherent to one another at multiple points so that the force generated to one muscle fiber can be transmitted to the next. This type is found in the walls of the gut, bile ducts, ureters, uterus, and many blood vessels. In terms of physiohistological structure, the smooth muscles are quite distinct. Smooth muscles contain gap junctions. The gap junctions facilitate cellular communications, so the smooth muscles can contract in a coordinated function. Second, in smooth muscles, there are no Z discs. Instead, they are replaced by dense bodies or areas which facilitates coordination in contraction. These bodies are attached to each other by intermediate filaments which contain the proteins desmin and vimentin. And third, it also has cavuli, a remnant of the T-tubules. It is rudimentary since the sarcolemma communicates with the sarcoplasmic reticulum via the secondary messengers. The contraction of smooth muscles is controlled by hormones or neurotransmitters. The smooth muscle is innervated by the autonomic nervous system. The autonomic nerves that supply smooth muscles have a series of varicosities which contains vesicles for the neurotransmitters like epinephrine, norepinephrine, and acetylcholine. The neurotransmitters, as well as the hormones released from the bloodstream, affects the activity of smooth muscles. Contraction of smooth muscle requires the phosphorylation of myosin light chain. The phosphorylation occurs in response to a rise in intracellular calcium either after an action potential or in the presence of a hormone or agonist. The regulation of myoplasmic calcium involves the sarcoplasmic reticulum and the sarcolemma. In the sarcoplasmic reticulum, or the SR, calcium enters the cytoplasm through channels located in the cavuli. Next, calcium release from the SR can occur either via calcium-induced calcium release and, more importantly, via inositol triphosphate or IP3 activation of SR calcium channels. In the sarcolemma, there are channels like sodium-calcium exchangers which allows the influx of one calcium ion for every efflux of 3 sodium ions. The rise in intracellular calcium in smooth muscle results in the binding of 4 calcium ions to the protein calmodulin. Calcium calmodulin complex activates MLCK, forming the CCMK. CCMK phosphorylates the regulatory light chain of myosin, or MLC, at the expense of ATP. Depending on the phosphorylation state of the attached MLC, the cross-bridge cycle velocity can be rapid or slow. In rapid velocity, it requires high myoplasmic calcium, therefore maintaining the myosin light chain cross bridges phosphorylated. It has 4 steps. S1. In the relaxed state, cross bridges are present as a high-energy myosin ADP-PI complex in the presence of ATP. S2. The non-attachment of the phosphorylated myosin to the actin filament will happen, producing a 90-degree angle, and there will be a release of ADP and PI from the myosin head. S3. This attachment will create a ratchet action by pulling the thin filament towards the center of the thick filament and generate a force, creating a 45-degree angle. S4. Release of ADP and PI will allow attachment of ATP. ATP decreases the affinity of myosin for actin, allowing the release of myosin from actin. Energy from the newly bound ATP is then used to produce a conformational change in the myosin head, so the cross bridge is again ready for another contraction cycle. Slow velocity. There are times when there is low amount of intracellular calcium but still above the threshold for contraction. The attached cross bridge is dephosphorylated by myosin phosphatase, thus the rate of cross bridge cycling is decreased. S1 is the same with that of the rapid velocity, which is phosphorylation of the myosin light chain by MLCK and ATP, which are produced by activation of calcium calmodulin complex. S2. Next, there will be again attachment of the myosin head to the actin filament, producing a 90-degree angle. In addition to the release of ADP and PI in rapid velocity, in slow velocity, there will be dephosphorylation of the myosin light chain by myosin phosphatase, producing a phenomenon known as the latch state. Latch state refers to a state in tonic contraction during which force is maintained at a low energy expenditure. This latch state will cause the longer duration of slow velocity contraction coupling. S3. After which, there will again be a production of the ratchet action, producing the 45-degree angle, which will pull the thin filament towards the center of the thick filament, generating a force. In this case, it will take a longer duration and an increase in force-generating conformation. S4. Attachment of ATP to the myosin head will also produce its detachment from actin. This will prepare the myosin for recycling. But in slow velocity, reattachment of the phosphate is needed before recycling, and prior to this, cross-bridging in a relaxed state occurs. In intestinal spasms, there is an abnormal intestinal smooth muscle contraction. This may be caused by irritable bowel syndromes. In the treatment of intestinal spasms, antispasmodic drugs such as hyoscyamine and dicyclomine are used. Antispasmodic drugs relax smooth muscles of the gut by blocking the actions of acetylcholine of parasympathetic sites of the smooth muscle, secretory glands, and central nervous system. In hypertension, there is an increased pressure in the blood vessels. Therefore, there is less space for the blood to travel through. In the treatment of hypertension, calcium channel blockers such as nifedipine are used to inhibit the entry of calcium to the myoplasm. Thus, there is a decrease in contraction, blood vessel dilation, reduced peripheral resistance, and decrease in the blood pressure.